we'll get started. Today is week 5, and we'll be talking about the RNA-Seq bioinformatics pipeline development. If everyone could please go to the address that I have down on the title slide and fill out the poll. I have too many use cases to talk about today, so I'm gonna let you guys pick, and whichever one you pick, we'll talk about, and then I'll get through as many as possible. But I didn't want to overwhelm with use cases. So while everyone is looking at that, just to review, this is just the basic next generation sequencing pipeline. When I spoke 2 weeks ago, we talked about template preparation a bit and the actual sequencing. Today we're going to be talking about the bioinformatics analysis. So an important thing with any bioinformatics analysis and any experiment really is to know what your biological question is. The experimental systems, your high throughput technologies, the bioinformatics and mathematical modeling techniques you use on your data are all interconnected. And you have to take them all into account when you're developing a pipeline or an experiment in general. So I always like to make sure we know what our biological question is. And with that, I just wanted to ask everyone what you wanted to learn about. So if you go to the poll down at the bottom, and I'll type it. Can you type it in the chat, Bill?
Yes.
And so I have 4 different presentations on use cases that are up. I'll go through the most popular one, which looks like it currently is the HPV use case. And then I'll go through them in order of popularity until it's the end. And if anyone wants to stay online after, I'm happy to go through additional use cases that we don't get to today. So just let me know in the chat. Also, everyone on chat, please type your full name into the group chat so that for attendance purposes. All right. Okay, so you guys can fill out the poll. I'm going to move through the introductory slides a little bit quickly because a lot of it's quality control and somewhat boring to a lot of people. But it's very important that you understand everything that goes through processing sequencing data because it is a lot. So the first thing is just sequencing terminology if you're not familiar. So we have our large DNA molecule that's broken up into itty-bitty pieces, and this is called the library. So this is your physical molecule. Then that's put through the sequencer, and it is transformed into a digital sequence of nucleotides or bases, and those we term reads. Then each individual reads, it's kind of like a puzzle in a box. You have to figure out where each of those pieces goes on your genome that you're sequencing, and that's termed alignment. Each of these reads also has a read length, which we talked about a few weeks ago, and that's just the number of bases that the sequencer was able to capture. Another set of terms that are important for sequencing is coverage. There's 2 types of coverage. You have your depth of coverage, which given a genome, so this is your genome, given any base pair on the genome, how many reads actually map to that position. So that's your depth of coverage. And that's referred to as like 60x. So that means there's 60 reads that align to a given base pair position on the genome. Breadth of coverage goes in the other direction. That's the percentage of bases in your genome that are covered by at least 1 read or at least 60 reads. So you'll often find that in papers, you'll see 80% coverage at 60x, which means that 80% of the target genome was covered by at least 60 reads. And sometimes this number can be the mean or the maximum or the minimum. So you sometimes need to pay attention to that because it is like maximum 60 reads. Then there may be a minimum of 1, which could be problematic for some applications. So there's two main types of sequencing that I just wanted to point out. You have DNA sequencing, which is whole genome and whole exome. This is actually sequencing the DNA molecule. And then you have RNA sequencing, which is that next step where the DNA is translated into RNA. And a lot of RNA sequencing focuses on capturing messenger RNA, which is then translated into protein. But you can also capture many different types of RNA, such as transfer RNA, small RNAs, microRNAs, and that kind of thing. And here, just note that a gene can have many different isoforms. So that just means these exons can be put together in a different order to create a slightly different protein. And so when we're aligning stuff to, when we're using RNA, the reads are actually coming off of the RNA molecule and maybe missing these intron sequences in the actual DNA. Okay, so this is the basic generic pipeline that I present. We're going to go through the first 5 steps, and then we'll get to the use cases that you guys have picked. So the first 2 are actually based around quality control, and this is very important to ensure that you have good quality reads, because if you have garbage in, you're going to get garbage out. So we want to make sure our data is good quality before we run it through our pipeline. The first step is just to look at the quality of the reads that are coming out of the sequencer. And I use a tool called FastQC. So each base that is read from the sequencer has a quality score associated with it. And this plot is the main plot I use in FastQC that just lists the length base pair of each read, So these reads are actually 40 base pairs long. And then these are the quality scores mapped out as a box plot with the quality score. So higher score means it's got higher confidence that that base was what it called. So here we can see we have good quality at the start of the read, but whoops, it gets worse, poor quality towards the end of the read. And again, that's caused by sequencers where there's dephasing that goes on. The longer the read gets, the poorer the quality. So you usually see this trend a lot with sequencers. And we talked a bit about base call quality from week 2, if you were here for week 2. So this is just an example of a good FastQC plot where all of our base pairs are nicely up in the green, good quality scores. So if you see something like this, what you're going to want to do is actually trim these off using a tool called Trim-O-Matic. And I didn't put a slide in here because there's not much to the trimming. It's just you're trimming off the bad base pairs of the reads, because if you have poor quality calls, it's not going to align very well to the genome. So after we do our quality control, we then go into alignment. So there's 2 types of, I guess there's alignment, and then you may have heard of a term called de novo assembly. Alignment actually uses a reference genome, so you know what the genome is. It's like having the picture on the box for a puzzle. You know what the picture is. De novo assembly is you have all of the pieces, but you don't know what the picture is. You don't have a reference genome. So de novo assembly is a bit more computationally intensive, and I've tried to do it, but it crashed my computer, so I haven't tried to do it since. But mostly what I use is a reference genome, and you know what that genome is, so you align all of your reads to that genome. So alignment tools have some challenges. There are different types of alignment tools. For a lot of high throughput technologies like Illumina, you're going to be using a short read aligner. There are also long read aligners for technologies that have very long reads that are in the thousands of base pairs per read. Generally, we're using short read aligners, and you have the ones in here in red aligned to the DNA. The ones in blue are splice-aware aligners, and they align to the DNA, but they're aware that there are splicing junctions. And I'll show you how that works in just a second. Some computational challenges are referred to as the read mapping problem. The shorter the read is, the less likely it is you're going to find a unique position on the genome for where that read goes, so you prefer longer reads. Repetitive regions are very hard to map to. And reads that have sequencing errors, so again, we want to trim off those poor quality bases, because if there's sequencing errors, then it's either going to align to the wrong position or not align at all, and you're losing data. And then sometimes the orientation of the read to the reference genome, whether it was on the positive strand or the negative strand, isn't known, so the aligners have to take that into account as well. And then RNA-Seq has the additional challenge of splicing junctions.
Amy, one question. Yeah. If you have microRNAs, these are like 25 nucleotides. Do you think at this instance, like RNA-Seq won't be a good idea?
For microRNAs, well, since you're sequencing the microRNA, it is RNA-Seq, but I think there are special methods that do the mapping for microRNAs.
The length is like 25 nucleotides. So
yeah, so they're really short. Yeah. So there's additional challenges with that. Okay. I haven't aligned microRNA sequencing data before, but I'm positive there are specialized tools out there for that. Yeah, that when you're measuring like really short RNAs, it's challenging. But, I know there's an area out there that does that. So if you need help finding a tool or anything, okay, I'd be happy to learn. All right, so in general, I used to use Top Hat 2, which is a splice-aware aligner. I now use STAR, but I like explaining Top Hat 2 because it's a little easier to understand than the STAR algorithm. So what Top Hat 2 does is it uses a DNA aligner first, bowtie 2, where it takes all of the reads, these orange things and the blue bars here, and it tries to map them to the DNA. So it's trying to map to the gene region that includes introns. It's not aware of splicing junctions, so any read that actually spans a splicing junction is not going to be able to be mapped with bowtie 2. So you'll see it'll map everything neatly inside of exons that don't cross splicing junctions. Everything with a splicing junction is going to be left as an unmapped read. Then Top Hat 2 runs an additional algorithm on the unmapped reads, and it identifies the splicing junctions because it's splicing aware. And what Top Hat 2 can do is identify known splicing junctions, but it can also identify novel splicing junctions. And sometimes this is where paired ends come into play. So remember we talked about from week 2, paired end sequencing is where you sequence the read from one end, or you sequence the fragment from one end, and then you also sequence it from the other end. And then you have this insert size, which is the size of your fragment. Aligners like Top Hat 2 actually take this size into account in order to align those two paired ends correctly. And so just a note, if you are using Top Hat 2 or any aligner really, check the default parameters, because sometimes Top Hat 2 has a default parameter for your inner distance, which is what it needs to calculate. The inner distance is just your insert size minus 2 times your read length, so that part that's in between the reads that you didn't sequence. So some aligners add a default value in, and other aligners don't, but you want to get that value probably from the sequencing facility or whoever prepared your library, and put that into your aligner so that it can align those paired ends correctly. Okay, after alignment, then we have our FASTQ file that is now transformed into a BAM file, and then we have to do some pre-processing on BAM files. So this is like a mini workflow in itself. It's very project dependent as far as what you need to do with it, what aligner you used, that kind of thing. So a BAM file contains the read name and the read sequence, as well as the read quality scores, just like the FASTQ file did. But it also contains the alignment information, so which chromosome it aligned to and what position on that chromosome it aligned to. and it contains other information. One of the most important ones to know about is the SAM flag. So there's a tool called Explain SAM flags, and these are just numerical flags that give you a bunch of information about that particular read. For example, a flag of 4 means that read was unmapped. It wasn't able to find the location on the genome. You can also identify if the mate is unmapped, or if the read mapped to the reverse strand. And you can also identify if the read is paired or unpaired. So there's a lot of information you can get from these flags, and this tool actually can help you interpret that numeric flag. So a lot of times in pipelines, I can extract out the unmapped reads because that gives me less data to work with if I'm not needing the unmapped reads, or if I only want to work with the unmapped reads. And another important thing to know when you're working with BAM files is you always need to know what reference file or what reference genome and version it was aligned to. For example, it's not enough just to know it was aligned to human. You need to know the version of the human genome, because version to version, the gene coordinates change. So if you don't know what reference genome and version your BAM file was aligned to, all of that alignment information is useless. Okay, so once you get a BAM file, sometimes there's some additional processing that you need to do with your BAM file before you can move to your downstream analyses. Tools such as SAM tools and Picard tools do a lot of these functions really well, and they're very well maintained tools. I use SAM tools like all the time when I'm working with BAM files. So this is just an example of pipeline. Sometimes you might want to filter your reads, but not always. For example, if it was aligned to a human genome and you're looking for viral reads, you're going to want to filter out the unmapped reads and align those to a viral genome. You generally always need to have your reads sorted either by name or coordinate for downstream tools to be able to navigate the file easily. And then sometimes you're going to want to mark duplicates. So back in week 2, we talked about PCR amplification. And this is where taking that information into account comes into play. So if you're doing a variant analysis, you need to mark and remove those PCR duplicates at this phase before you move forward with your variant analysis. But if you're doing RNA-Seq gene expression, the recommendation currently is that you don't mark or remove duplicates because that can remove actual biological signal. Then you can also add and remove read groups. This helps if you have the same sample separated across multiple lanes and you need to merge the information at some point into one file. You can actually mark which lane each read came from in case there are difficulties downstream. And you need to identify which lane a particular read came from. And then you can merge multiple BAM files if needed, or that may not be needed if you haven't run technical replicates in different lanes. And then finally, the last step is generally always, downstream tools always require you to index your BAM file. And this is kind of like just a roadmap. So these tools can navigate these files more efficiently because BAM files can be rather large. I like to stress this slide because up until BAM preprocessing, most sequencing, DNA-Seq, RNA-Seq, whatever, most sequencing pipelines are very similar up until the BAM preprocessing stage. And BAM files are usually the files that you will find on public databases, such as the Genomic Data Commons with the Cancer Genome Atlas data. They have BAM files that are already aligned to a human genome because they're smaller than FASTQ files. So with BAM files, you can go in a lot of different directions. You can filter your reads so you can reduce the file size so that it's more manageable. You can convert your BAM files back to FASTQ, which essentially just takes out all of the alignment information and gives you the read and its quality so you can start the pipeline over again. You can also do variant analysis, differential expression, and you can do visualization on BAM files. So, and there are certain tools like the BAM2 Fast the BAM2 FASTQ script is what converts BAM files back to FASTQ if needed. So, for example, with the viral example, I take out all the unmapped reads that didn't map to human, and that would create a separate, smaller BAM file, and I'd convert that back to FASTQ and then align to the viral genome, which is much, much, much faster than converting the entire human mapped file back to FASTQ and then mapping to viral, because there's so much human data in there that you really don't need to process. Okay, with RNA-Seq data, generally a lot of pipelines will go through the getting count data for your genes or whatever features that you are trying to figure out what the expression is. So with getting count data, first you need to define what your features are. What are you interested in counting? Are you interested in counting genes, exons, transcripts? These are all different features that you can define in what's called a GFF or GTF file. And those files just annotate your genome as to where, say, a gene starts and ends, or where an exon starts and ends. And then the programs that do the read counting will, any read that maps to those coordinates, it will increment the counter for that feature. So feature files from well-known organisms can be downloaded from the UCSC table browser and GenCode. I get most of my feature files in reference genomes from GenCode. So what some of these algorithms do, you'll notice I have up here genes and transcripts. So there's a difference. So transcripts, counting features for transcripts actually differentiates between isoforms. And you need special tools to actually figure out if a read goes to one transcript or another. For example, here we have two isoforms of the same gene. So here's the gene on the genome. You have isoform A with 3 exons and isoform B that only includes the first and the third exon. Any read that maps to the second exon, we know is a count towards the isoform A. Any read that crosses the splicing junction between 1 and 3, so these blue ones up here, we know for sure belongs to isoform B. However, these purple reads that map cleanly within isoform A, or within exon 1 and exon 3, we don't know, does that belong to isoform A or isoform B? So the algorithms, such as Salmon and RSEM, are 2 examples, will use the distribution of reads that you know belong to one or the other, and they will estimate the proportion of these reads that go to isoform A and isoform B. So once you do your feature counting, you'll end up with a count matrix. And so you have your genes down the rows, and then your columns are your conditions or your samples. So here I just have an example where I have 2 control conditions, and I have 3 experimental conditions. And I just want to point out just a few properties of RNA-Seq data that are different from microarray data if you're used to dealing with microarray data. The first thing you'll notice is that these are discrete counts. There's no 0.567 or anything like that. They're discrete counts. 1 read equals 1 count. And then you'll also notice that there are a lot of zeros. And this is just a small sample that I manipulated, honestly, from actual data. But the real matrix contains, like, mostly zeros. But the zeros here actually mean something. It means there was, it found no actual fragments of that gene in that sample at all. So there's a biological meaning to these zeros. So we can't just normalize them out or get rid of them. Also, we'll see in a little bit that low read counts, such as you just have 2 reads that align, can actually cause exaggerated fold changes when you're doing differential expression. So once you get your read count matrix, you can go in 2 directions. One, you can feed your raw read counts into differential expression, which we'll see in a moment. Or you can calculate an RPKM or FPKM or TPM value, which these are calculated if you're just looking at the absolute gene expression, so it's not relative to anything. And the reason why you want to calculate these is because each gene is a different length, so you need to normalize for the size of a gene. For example, if a gene is 10 base pairs and it has 10 reads aligned to it, versus a gene that's 100 base pairs and it has 10 reads aligned to it, you're probably going to have 10 full functional transcripts of the shorter gene versus 1 full functional transcript of the longer gene. So you have to normalize for that gene length, because comparing a 10 to a 10 doesn't mean the same thing if you're comparing across genes that are different size. Also, you need to normalize for your library size. So that is just the total number of reads that have been counted for a particular sample. If a sample only captures, say, 10 reads, and then another sample captures 100 reads, and a gene is expressed 10% in both of those, you're only going to identify one transcript in your smaller library and 10 transcripts in your larger library. So you have to be able to normalize that difference to be able to better compare across samples. So the RPKM and FPKM is just reads or fragments per kilobase per million. FPKM is used for paired end reads. In order for the counting program to know to count fragments correctly, it has to know that the data was generated with paired ends. If you don't tell it that it's paired ends, it's going to think it's single ends and double count a lot of things. So that's one important setting to know when you're dealing with some of these counting programs. So with RPKM, FPKM, it normalizes for the sequencing depth or the library size, and then normalizes for gene lengths. However, this leaves that these expression values are not really directly comparable across samples. The transcripts per million is becoming more and more popular in the literature, and it uses the same data, but it does it in the reverse order. So it normalizes for gene length and then for sequencing depth. And this, well, a lot of people say, or some people say it's still not comparable across samples. I think it's more comparable because it more looks like it's a proportion of transcripts present in a sample. So you can't interpret it as a direct expression value. It's more of a proportion. Okay, use case. All right. So here are the results of our survey. So you guys want to learn about the HPV head and neck cancer patients from TCGA. And then if I have time, I'll go into this last one, which is a little short. So thank you guys for voting. I hope that was fun. I've not done that before. Okay, use case 1. Whoops. Guess that'll go away. Okay, so for our first use case, just a little bit of background about what this use case is and why we were pursuing it. We were looking at HPV infection in patients with head and neck cancer. HPV is highly correlated with cervical cancer in women. Over 80 or 90% of cervical cancers are thought to be caused by HPV infection. However, recent research has shown that in men, head and neck cancers, over 70% of head and neck cancers are thought to be caused by HPV. One of the clinical observations, this work was done in collaboration with Dr. Ian Morgan in the School of Dentistry. And their observation in the clinic was that patients that were found to be HPV positive in head and neck cancers tended to respond to treatment better than patients that were HPV negative. So their goal with this research was to analyze the data in the Cancer Genome Atlas and see if we could come up with a biomarker for HPV positive head and neck cancer patients, because the biomarker that they use for cervical cancer doesn't respond the same and is not as consistent in head and neck cancers. Down here we also see that the overall survival, this is survival of patients in TCGA. Those that were HPV positive had a better overall survival than those that were HPV negative. So the first approach to trying to answer this question was to do some exploratory analysis and identify which human genes are differentially expressed in HPV positive versus HPV negative head and neck cancer patients. This is the cervical model of HPV. I'll just go over it briefly. So HPV infects the cell as an episome, and it's got these E6, E7 genes in there. It also has an E2 gene. While the virus is in its episomal form, the E2 gene expression suppresses the E6, E7 gene expression. However, at some point, the virus actually splices or cuts out the E6, E7 gene region and inserts it into the human genome. So it breaks the human genome, the human DNA, and it inserts its own DNA in there. So now the human transcriptional machinery is transcribing the E6, E7 gene, and not the virus. So once that happens, you now have E6, E7 in your genome. While your immune system may actually get rid of all of the episomal HPV that's still left in your system, once it does that, the E2 gene is no longer there to suppress E6, E7. So once E6 and E7 become active, they inhibit genes like TP53, which is a tumor suppressor, and affects the cell cycle. So now the cell cycle is no longer controlled. And you have uncontrolled growth, and that's how the HPV kind of causes cancer in cervical. So we think with head and neck, it's very similar, but it's non-exact. So usually they stain for a P16 protein, that's a human protein that's upregulated in cervical cancer when they're infected with HPV. However, head and neck cancers tend to have higher regulation of this protein or lower regulation, so it's not directly correlated as closely with head and neck cancer. So the gold standard for identifying HPV positive is to look for this E6, E7 gene expression in RNA-Seq data. So this is just a little bit about the Cancer Genome Atlas. It was a coordinated effort to accelerate the understanding and molecular basis of cancer. They have over 11,000 patients with GDC now. I think that's up to 30,000 patients maybe. But it's rapidly growing, and the cool thing about TCGA is that for each patient, you have the tumor and you also have a matched normal tumor. And then each sample was run on a bunch of different modalities. So you have RNA-Seq, whole genome sequencing, microRNAs, protein abundance data. And so you're able to take all of these different platforms and for the same tumor and the same sample and compare. So with this data, we had 520 head and neck cases that were available. These were pre-aligned to the human genome, so we're dealing with BAM files. The first problem we needed to solve was which patients were HPV positive. So the solution to that was to classify the patients as HPV positive or negative, and then perform our differential expression analysis. So here's the modified pipeline from the general that I did for this data set. We didn't need to do the first 3 steps. They were already done for us. So I started with BAM files, but they were already aligned to the human genome, and I needed to look for viral E6, E7 expression. So what I had to do is, starting from the BAM file, modify the pipeline. So the first step was to filter those aligned BAM files and extract out all of the unmapped reads. And then I used the BAM to FASTQ script to convert those unmapped reads back to a FASTQ file. And then I aligned that FASTQ file to the HPV reference genomes, and I was able to align to, there's like almost 200 or over 200 at this point. But I created a reference genome with all HPV genomes in it. So I was able to align to all HPV genomes at the same time. so we could figure out which patients were HPV-16 positive versus HPV-18 versus on a non-cancer causing HPV late 33. So once the alignment was done, I had the annotated genomes for the HPV reference genome, where all the genes were located. And so we got the count data using HTC count. And then once we got that count data, we didn't have to do anything to it. All we did was, if there's a read count of 10 or more for E6 or E7, we counted that patient as HPV positive. And through this method, we were able to classify the patients as HPV positive or negative. And we actually found that the TCGA P16 staining classifications misclassified about 10% of the patients. So now that this pipeline was just getting a classification. So we were looking at viral gene expression. Now we want to shift back to looking at human gene expression now that we have our patient classifications. So here, again, we start with the aligned BAM file that's aligned to human already. We didn't need to do anything with it. It was already processed. And so we just had to find the GFF file for that human genome, which in this instance was HG19 at the time. So we got the matching annotation file, fed it into HTC count, and counted up the reads for the human genes. And then we took that count data and went and did a differential expression analysis. So this is just a slide you've already seen. This is the count data. It's the same example data. I just put the headings up here. So now we have our controls. We use as HPV negative patients were our controls because we wanted to see which genes were up or down regulated when the patient becomes infected with HPV. So here we have our HPV positive patients over here. So I had talked a little bit previously about the many zeros and the discrete counts for RNA-Seq data. That actually leads to this data having a drastically different distribution compared to microarray data, which is continuous. So microarray data is actually based off of the fluorescent intensities off the chip. So you're never going to get a reading of 0. There's always going to be a background signal intensity, and it's going to be a continuous measurement. With RNA-Seq data, you get a large number of zeros in your data, followed by additional expression values. So you can see RNA-Seq data is a bimodal distribution, whereas microarray data is more similar to a normal distribution, not exactly, but it's more similar. So a lot of microarray normalization methods that are used actually assume a normal distribution, but you can't assume that with RNA-Seq data, obviously, because it's nowhere near normal. So when you're dealing with RNA-Seq data, you just got to keep in mind that there are special methods that you have to use that are specifically designed for RNA-Seq when you're doing differential expression or any type of normalization for this data. So 2 packages that I've used are DSeq2 and EdgeR. I prefer to use the DSeq2 package because it controls for exaggerated fold changes due to low read counts. For example, this is just a small table I have here. Both gene 1 and gene 2 are calculated with a fold change of 2, but gene 1 only had 1 read aligned to the control, and gene 2 had 2 reads in the experiment, whereas gene 2 had 100 to 200. So this is a much more robust and believable fold change of 2, whereas this is probably just noise. So DESeq2 actually accounts for this and adjusts fold changes and p-values accordingly. The nice thing about both of these packages is you don't have to worry about TPM or normalization. You just take the raw read counts from your counter and you put them into this program. And this is just an example output of DESeq2 where you'll see the base mean, which is just the average across all samples for a particular gene, as well as the log 2 fold change and the p-value and adjusted p-value. When you're doing gene expression, always use the adjusted p-value because it corrects for multiple testing. So with RNA-seq data, filtering, this is based off of fold change, level, and p-value. I'll usually use the adjusted p-value, the fold change, as well as a base mean filter. So you can have a large fold change and a significant adjusted p-value, but a very low base mean. So anything with a base mean below 10, I usually just get rid of off the bat, regardless of p-value. And this is just an example comparing DESeq2 to EDGE-R, where you can see there, here you have 3 controls, and these are the 3 experimental conditions. DESeq2, these are all really low read counts. DESeq2 gives it a negative 1.6 and does not even bother to calculate a p-value, whereas EDGE-R gives it a negative 6 fold change with a very significant p-value. This is a gene that I would probably disregard in any analysis, which is why I prefer to go with the DESeq2 measurements. However, when the read counts are high and you have a base mean, the fold changes for DESeq2 and EDGE-R and the p-values are very comparable and comparable. So I always prefer to use DESeq2, but DESeq2 and EDGE-R do make different assumptions. So depending on which assumptions you are more comfortable with, and if you don't like the extra statistical manipulation of data with low base means, then you might prefer to use EDGE-R. But both are used rather frequently in the literature. So the DESeq2 output for this experiment actually gave us close to 3,000 differentially expressed genes with an adjusted p-value of less than 0.001. And so you can manually look through the genes, or you can cluster them if you have a time course or you have multiple samples. But the one thing that we did with this set was we just wanted to know what's there, what pathways and biological processes were represented, and was it what we expected in when you're looking at a patient that's become infected with a virus. So this leads us to the enrichment analysis, which you will hear more about with Dr. Dosmarov's talk coming up in a week or two. He'll go into this in a little more detail. So the tool that I used was David, where you just have to, it's just an online tool. You input the gene symbol of your gene list that you want to do the enrichment analysis on, and then it'll, it has a tool on there called functional annotation clustering, which I really like a lot. So what I did with David is I took all of the genes that were significantly downregulated as one group and all of the genes that were significantly upregulated as another group and input those into David as 2 different lists. So our downregulated genes had functions like epithelial cell differentiation, cell migration, cell adhesion, and an enriched pathway was focal adhesion. Our upregulated genes included DNA replication, DNA repair, cell cycle. The pathways were mitotic cell cycle and DNA replication pathways were all upregulated. And all of this makes sense because, again, when you're infected with HPV, it is breaking your DNA and inserting itself, so you're going to need that DNA repair. And it's also having an effect on the cell cycle. So that told us that the differential expression analysis and the gene list that we got makes biological sense. So some of these methods were published back in 2017 if you're interested in reading more about it, or I'm happy to answer any questions about it. If anybody has any questions about this use case, you can ask now, and we have just a few more minutes. I can quickly run through another one. And if anyone wants to stay on, there's nobody in this room after, I'm happy to just keep going, so just let me know. Okay. All right, so the next most popular use case is a knockout experiment effective if there's measured gene expression for a knockout? I like this one. This one was fun. So that's use case 4. This one's a little short, so it shouldn't take too long to go through. But if anybody needs to leave, feel free. So this is actually a use case with that was done on the plant Arabidopsis thaliana. So we were looking at trying to figure out transcriptional pathways in the Arabidopsis roots. So specifically, we're looking at ARF19 signaling. From some previous microarray experiments that we did, we narrowed down on ARF19. It's a known, it's the auxin hormone affects growth through ARF19. So we wanted to figure out what genes ARF19 was actually controlling. So we actually knocked ARF19 expression out and wanted to see how the gene expression program was changed. So here we have our, this is just our the read count matrix that you guys saw before. And instead of HPV positive and negative, we now have the control, and then we have the auxin induced. So for this pipeline, we essentially did all the quality control. I did the BAM preprocessing, and then I did the count data, and I did a differential expression analysis, which we just went through that pipeline, so I'm not going to go through it again. But what we found was some oddities in the expression values for ARF19. So we used visualization of the BAM file of the actual aligned reads to figure out what was going on. We already talked about these slides, so I will skip them. Okay, so after a differential expression analysis, for this experiment, we had done a microarray experiment with the wild type, so it had ARF19 in it. So the ARF19 gene expression was of around 3.7 in the microarray data, and then we did the exact same experiment using RNA-Seq in a pilot study. We were trying to see if RNA-Seq correlated well with microarrays, and it does really well. And the expression of ARF19 was 3, which is rather comparable to microarray. And then we did the ARF19 knockout and did the RNA-Seq analysis on an ARF19 knockout. So this meant that ARF19 should not be expressed. It's not there. We knocked it out. It should be gone. But when I did the differential expression analysis, I found, oh, we actually have, this was statistically significant too. We had significant expression of ARF19. So our question was, well, if this gene was knocked out, why do we have expression? So to figure that out, I took the BAM file of these samples and imported it into IGB, the Integrated Genome Browser, and calculated the coverage depth of coverage graphs. So the height of the graph here indicates how many reads actually align to that position. So this goes up to around 250. Both of these are at about 250 reads per position. So you can see these are the exons, or the introns, where no reads aligned, or they aligned across the splicing junction. This gene, ARF19, is actually on the negative strand, so it's reverse transcribed in this direction. So we find in the wild type, the entire transcript is expressed rather robustly. However, in the ARF19 knockout, we have expression at the start of the gene, at the start of the transcript, but then there's like almost no expression down the rest of the transcript. I asked them how they did the knockout, because I thought this was really odd, and they said it was an insertional knockout. I found out that the insertion point was actually right here after this third exon, where they inserted a section of DNA that would knock the transcriptional machinery off the transcript, and so that it couldn't finish processing, transcribing that mRNA. And so essentially I had the PI look at this, and it's trying to express this gene, it's trying to transcribe it, so you get a lot of reads at the beginning here, but then it gets knocked off and it really can't finish it. So the conclusion here was, and I had the biologists investigate this, is this actually a functional transcript? Is it doing anything in the cell? And the conclusion from the research was, no, this is not a functional transcript. So we can say with confidence now that our knockout procedure actually did work. So this was just a way to, we got some odd data, we're like, why? And because we did RNA-Seq, we were able to actually go in and look at exactly what part of the transcript was being expressed. If it had been a microarray experiment, we may have still seen expression value there, but we would have not been able to dig down into the details to see what part of the gene was being expressed and to be able to come to the conclusion that it was a non-functional transcript. So this is just a great example of how RNA-Seq can give you a lot more information, especially when you need to go investigate a problem. So,
Amy,
yeah.
Given this data and giving it to the biologist, would you then, I mean, to make it cleaner, you'd want that insertion point prior to any initiation of the transcript. I mean, they say it's non-functional, but you don't really know what else it could be doing in there.
Right, and that was my question.
Yeah. So I mean,
But
ideally, wouldn't you want it at the?
I would assume yes, but it all depends on what is actually physically possible for that particular region of the genome. So, and I know in plant biology, I mean, they have their methods that are well vetted and everybody uses them, and this was just a surprising side effect, I guess.
Example of that.
Yeah, yeah. So you can see that there is some expression down here, so it didn't knock out everything, but it's so low that they think it really wasn't a functional protein that was doing much in the cell because of how high it is normally expressed. So, yeah, and they went and they looked at that and did some research on it, and was able to come to the conclusion that our experiment was good, the knockout procedure did work, so we didn't have to redo the experiment, which was great. Saves money, right? So, all right, so I think I'm a bit over time, so I'll stop there for a few minutes. If anyone wants to stay on, please just sit here for those of you here, and or you can text in the chat window if you'd like to stay on and look at some of the other use cases. Oh, and this was the microarray experiment was actually published that I just talked about several years ago, and we just got a new grant funded to do further RNA-Seq experiments. That RNA-Seq was just a pilot study, so we only did one time point. This is actually a time course for trying to identify signaling pathways due to auxin stimulus in roots. So hopefully we'll have more manuscripts coming out in the future. All right, I am gonna move. I'm just gonna keep going. So if you guys need to leave, just let me know. Anyone online, if you need to leave, go right ahead. I'm still recording, so if you have to leave and want to watch later, you can watch the recording. Let's see. So just so you guys here can see, the second most popular use case was what is the level of mouse genome contamination in human breast cancer patient-derived xenograft samples. This is a rather new project that I just started working on last year, and it is a really cool pipeline. I enjoyed doing this project. And this is a little bit longer, but nobody liked number 3, man. All right, that's okay. It's in the paper that we have being reviewed right now, so. Okay. So just a really quick high-level explanation of what a patient-derived xenograft is, because I did not know what this is when I started on this project. So you'll forgive me if you know what this is and I'm not explaining it correctly. You're welcome to correct me or talk to me after if I explain something wrong. But my understanding of patient-derived xenograft is you have a patient with a cancer, you take out the tumor, and you take out and you purify the cancer cells, and then you graft them into mouse. So for this data, we're doing breast cancer. So they grafted it into the mouse mammary glands, and they grow the cancer in the mouse. And then there's you have to duplicate this a bunch of times to make sure that that cell line is stable and growing in the mouse. and then you can start to do experiments on it. So what PDX models are really great for is experimenting on human cancer while removing the human element. So you can test out drugs if there's, and try and figure out if a particular cancer is responsive to a certain class of drugs. That's one paper that we just got published this year, and any other number of experiments. So for this data, we're actually looking at metastases and trying to figure out where these breast tumors like to metastasize to and the expression changes that occurred when they did metastasize. However, this pipeline is focused on more of a quality control question. PDX data, when you take a tumor that's been growing in a mouse, it's human tumor, so that's human DNA. You're inevitably going to get mouse contamination. You're going to get mouse cells in there. And from my understanding, there are methods to try and separate those in the lab before you do sequencing, but they're not always perfect. So there was a recent study done that I do reference in the paper that was published that found out that digital sorting through alignment actually does the best in separating out the mouse and human genomic content. So this is a manuscript that was just submitted, and I have a slide at the end with the other one that actually did get published, which was a sub-study of this. But we did 119 PDX samples, and then we had 6 pure patient samples from VCU, and we had 9 normal mouse samples that we were looking at. We did RNA sequencing on all of the samples, and the question here, before we did anything else with this data, is what is the level of mouse genome contamination for each sample? And so the strategy was to calculate the % of reads that map to human and map to mouse. Some considerations that I had to take before starting this pipeline was that, one, we're starting from a raw FASTQ file, so all of the normal standard quality control steps that we've already been through, I had to go through with this. And then also human and mouse genomes are very identical. I found this 85% on the internet. I don't know if that's completely accurate, but I know some genes are more similar to human genes than other genes. So you can have a read from a mouse mapped to a human. And also the PDX samples we knew contained an unknown amount of human, mouse, and viral genomic content. So there was viral contamination in some of these samples as well. So here is the modified RNA-Seq pipeline that I did to answer this question. First we did quality control, and then I did some read trimming. And then before I did alignment, I manually created a custom reference genome. How I did this is I took the human genome, the FASTA file. It's just a text file. You can open it up in a text editor. But I used bash scripting on the command line to insert the prefix human or mouse before each of the chromosomes names. Because in human, the chromosomes are named CHR1, and in mouse, chromosome 1 is named CHR1. So if I just manually concatenated the FASTA files, I'd have 2 chromosome 1s, and I wouldn't know if it came from human or mouse. So I manually tagged human chromosomes and mouse chromosomes, and then just mashed the files together. The viral genomes, I also mashed in there, but they didn't have chromosome 1, 2, etc. So I didn't have to actually edit any of those chromosome names. And then I also did the same thing with the GTF and GFF file. Before I did like differential expression analyses, I had to go in and modify human and the human chromosome and mouse chromosome so that it would match to the chromosome name that's in the BAM file. And this is just an example of what the chromosome 1 looked like. So this is the name of the actual FASTA file header for chromosome 1 in the file that I used. And then this is the name of the mouse. So once I had my custom reference genome, I used the tool STAR to align these. STAR is much faster than TopHat 2. And it's also starting to be used a lot more. So I really like STAR. It gives you a lot of different outputs. It also has a very user-friendly manual for defining its like hundreds of options. And it also does some BAM pre-processing for you. So it will sort the BAM file for you, and it will, it does alignment, it does sorting, and I think it indexes the BAM files for you. So this actually saves you a bunch of pre-processing steps in your pipeline because STAR does it all for you. So the input to the alignment was the FASTQ file of a PDX sample, and then the custom human mouse viral FASTA reference genome. And the output was a BAM file that's aligned to human, mouse, and viral, and it does it all simultaneously. So the one important thing that I had to do with STAR and the parameters, STAR automatically will take one read and find multiple alignments and report multiple alignments. However, the alignments have a quality score, so you might get 2 or 3 different places that are read aligns with a different quality of alignment. This was a problem because it could align to human and mouse, but one of them is most likely going to have a better alignment score than the other. So I told STAR to give me the single best alignment for each read. So it would pick human or mouse. And then, like I just discussed, we don't need to do BAM pre-processing because STAR actually outputs all of that. It does all that manipulation for you, and really fast. So once I got the alignment, then I just had to go into the BAM file and get the species counts. Note that I don't use a read counter program here, and I didn't need a GFF file for this step. I just used the SAMTools program and a bash script that I wrote to go through the BAM file and count all the lines that mapped to, that had human prefix and all the lines that had mouse prefix. And that's what this data looked like. So I wrote a bash script to do this. And by the way, I'm happy to share any of my scripts with anyone. If you're trying to do something similar, let me know, and I'm more than happy to share the code that I've written to do some of these analyses. So here's just the sample name. So we have a PDX model that we've named HCI01. And this is actually a liver metastases from the breast cancer. We have lung metastases. These MGTs are the primary mammary gland tumors. And then we have some other samples that are more complicated that I won't explain, but they're in the paper. We have the different tissues. Here we have the total read count. So this is the total count of reads that aligned to my custom reference genome. Or no, this is the total number of reads, period. And then we have the total that aligned to mouse and human together. And then we have the total number of reads that just aligned to human, the total that just aligned to mouse. These other aligned are the viral reads. So you can see there is some differing viral contamination in some of these samples. And then this is the number of reads that are unmapped. So the percent of human and percent of mouse that I calculated for this data was based off of the total of mouse and human aligned reads. So we wanted the percent to add up to 100%. So we excluded any reads that aligned the viral, and we excluded unmapped reads, just to make the data easier to look at. Because you're always going to have some of these in there. And so you can see some of these samples have a different percent of human and mouse. For example, this one is 84% human. And this one is only 15% human, and it's mostly mouse. So this is the figure in the manuscript that is currently under review. I've redacted it from the slides that I've shared online, and I'll put this up there once the manuscript gets accepted. So this is just a bar chart that I created in R, and you have the percent here on the side, 0 to 100. You have the human is blue, the red is mouse, and they're sorted from highest human to lowest human. So here we can see that these are our 6 patient samples. They all have 100% human reads. And at this end of the spectrum, we have all of our mouse normal samples. they're 100% mouse. And in between, you have the different types of PDX samples that we extracted. I found it interesting that the mammary gland tumors tend to congregate towards this end where they're mostly human. So those are the primary mammary gland tumors grown in the mammary gland. Whereas the metastases, those that metastasize to other organs, actually started to degrade in their human content, where some of them are pretty close to not having any human at all. It's mostly mouse. So using this, we were able to filter out some of those samples that were severely contaminated with mouse so that we could focus on the human data. And Turner et al. Paper just got published this past March. It has some of these methods, including creating the custom reference genome described in detail in that paper. And then the figure that I just showed you is actually part of the Alzubi et al. Paper, which is currently under review by Breast Cancer Research. Any questions? Still have 7 people. All right. And then the last use case is actually a follow-up to this one. So I'll present that quickly if you guys want to sit and listen. Let's see. Is that okay? So I actually don't have to go through all these slides because we already did that. Okay. This one is a slightly longer pipeline, so this was probably the longest use case. But we already went through a bunch of it. So again, custom reference genome, we did the alignment the same way. We didn't need to do BAM pre-processing. And then it changes when we get down here to the species-specific read counts, because we're actually looking at gene expression at that point. So at this phase, so before, just to find those percentages, I was just looking at the BAM files. I was just manually processing the BAM files to figure out what aligned to human, what aligned to mouse. I didn't care where it aligned to human or where it aligned to mouse. So at this point, because we're actually, we're trying to get gene expression profiles, we're interested in which genes the reads align to. So for this step, I did use a read counter tool called Salmon, and then I processed that data in R. So the input was the BAM file. The output is a data matrix of human read counts mapped to genes, human genes, and a data matrix of mouse read counts mapped to mouse genes. And this is just an example of what that looked like for a few of the samples. So here we have our PDX models. We have a mammary gland tumor and then a metastases. And then we have just an example of a patient. These patients, we actually had 6 samples, but it was just 3 patients. So it's patients with the matched breast cancer that metastasized to the brain. So these are actually matched breast and brain metastases samples. And then we have normal mouse. So one thing with these read counts that I found cool, and again, quality control is what you try and make sure the data looks like what you expect. These top 4 genes here are the human genes, so the human T-span6, and then these are the mouse, the orthologous mouse genes, so this is the mouse T-span6. So you notice for the PDX samples, you actually have read counts mapping to the human gene and to the mouse gene. For the patient data, you only get expression in the human genes and nothing maps to the mouse genes. Except there's one little odd one here. Like I said, there is noise. So we did find a little tiny bit of patient expression in the mouse that got mapped to mouse, but it was really so small, it's what we expected to happen. However, if we would have gotten a lot of patient data mapping to mouse, we would have said, hold on, there's something wrong with our pipeline here. And then the reverse happened for the human genes, where the mouse, there was no mapping of mouse, the normal mouse to the human genes, and all of it happened mapped to the mouse genes. So again, this was consistent across all of our samples. So It's what we expected to happen, and it was just a quality control check to make sure that our pipeline and our aligner was doing what it's supposed to be doing. Okay, I've kind of shifted up the pipeline here. The first thing that we wanted to do is to, the main use case for this is to figure out which breast cancer subtypes our PDX models most resembled. Because the PI, Dr. Chuck Harrell, in the pathology lab here at VCU, he kind of knew what some of his PDX models were supposed to be HER2 or basal-like and that kind of thing. So we took the gene expression that we calculated from this pipeline and input it into a classifier to make sure that they actually got classified as what we expected them to be. So in order to do that, we took the read counts for the gene expression read counts, and then we didn't do a differential expression analysis at this point, although that is in the paper. If you're interested when it gets published, we calculated the transcripts per million because we're looking at absolute expression values here. And so I did that in R. I wrote a custom function to do it based off of the TPM equations that have been published. So the input is the human count matrix. So we're only looking at human gene expression at this point. I separated out human and mouse, so we only have human. And we're not looking at mouse gene expression. And the output was a human TPM matrix. And then this TPM data went into the R package GeneFu, which does breast cancer subtype classification. And I use the PAM50 model. So we're looking at the 50 genes defined in the PAM50 matrix. So subtype calling with GeneFu, use PAM50. GeneFu outputs the discrete calls as well as the probabilities for each subtype. So down here, I know it's a little small, I should have made the font bigger. Here we have the discrete calls for basal. So our HCI01 PDX models were classified as basal, which is what we expected. HCI08 was our one HER2 that we knew was HER2, and it got correctly classified as HER2. And then HCI09s we knew were luminals, and they were classified as luminal B. But you can also get the probabilities to see how close was that call, how confident was it that it was a basal or a HER2. So for the HCI01s, it was pretty confident that those were basal, over 50% confident. HCI08, it was kind of stuck between HER2 and luminal B, but HER2 won out pretty significantly with 54%. And then HCI09s were actually much closer between luminal A and luminal B. And there were some that were even closer than this, so it's just like a luck of the draw kind of thing, that it got classified as one or the other. But just a warning about this classifier is that depending on the number and type of samples that you input into the classifier, your classifications will change because it's based on that distribution of the samples that you input. So I think it's better to input more samples than fewer because it has more data to work with. It's also important to filter your samples before you go and classify. So I actually did this several times with a bunch of different subsets, and we ultimately ended up deciding to exclude all samples with less than 10 million mapped human reads. Because when you include mouse samples in there, it really screws up the classification. And if you include samples that have very few human reads and it's mostly mouse, it again messes up the classification. So we actually used the data from the previous use case to filter down our samples. So some of our samples actually don't have a subtype associated with them because we didn't have enough human expression data to do the classification. And then we took the the 50 genes that it used to do the classification and took their gene expression profiles for one PDX representative sample. Each of these have about 3 to 6 replicates, biological replicates. We just picked one. And we did a principal component analysis just to see where the main variability was. And it turned out that it very nicely clusters basal PDX samples with basal, indicating that their gene expression profiles for those 50 genes are highly similar. Our HCIO8 is kind of out here in the middle, and then we have our luminal Bs on this end of the spectrum. And then we have our one lim2, which is an odd sample that has some oddities with it, but it is a basal, so that's the only one that kind of sticks out. Are there any questions so far? Almost done. Okay, so we have our classification. So that answers our question that we were originally asking. That's our use case. But then we wanted to go and verify, do our gene expression profiles for these 50 genes that we use to classify, do the PDX model profiles actually correlate with pure real human data? So we wanted to take our gene expression profiles from the PDX models and integrate them with the TCGA patients who are also classified based off of subtype, and see if like the HER2 clusters with TCGA HER2 samples, if our basal samples cluster with TCGA basal samples, or do they not cluster? So this was another form of verification that we're getting what we expect to get. And our PDX models do appropriately represent actual human data. So to do that, we did I took the TPM data, and then I actually merged it with the TCGA data, which you can get TPM values from the TCGA data. They used RSM, I believe, to extract to calculate TPM values for TCGA data. So essentially, that's just merged together in a matrix, and then I needed to normalize and center that data. So this is some additional pre-processing because you're going to have batch effects from our PDX samples. The TCGA data was processed differently as PDX versus human, although we want to keep those differences. So we did batch effects to reduce, or we did this normalization to reduce batch effects, and then centering just helps view it on the heat map a little better. And so then we took those genes and just clustered them on the heat map to visualize the profiles. So this is the figure. It's a heat map. Your 50 genes are along the y-axis. Your patients are along the x-axis. We took our 101 PDX samples, again, because we filtered out those with less than 10 million human reads. So there are 101 PDX samples and 817 TCGA patients for breast cancer that were already subtyped. And so we went ahead and we took the gene expression of the PAM50 genes for our samples and the TCGA samples and clustered them using hierarchical clustering. So this annotation bar here tells you if it's a PDX or one of our samples or a TCGA sample. So all the grays are the TCGA samples and the black bars are our PDX samples. The top annotation bar tells you which PAM50 subtype it is. Dark blue and light blue are the luminal A and B. Pink is HER2 and basal is red and normal is green. So you can see that all of our basal-like PDX samples cluster nicely with the TCGA basal-likes. Our luminal A and B samples cluster with the TCGA luminal A and B samples. And our one single HER2 sample clusters with the TCGA HER2 samples. So with this verification, we were able to confidently say that our PDX models are similar to actual human data. So any observations or conclusions that we make off of our PDX models, such as drug response and that kind of thing, can possibly be more easily transferred over to actual human data. So, and this is the same slide that you saw last time. So those were my 4 use cases. Thank you guys for sticking around. And the recording's up there if you need to watch it, or for anyone who's who had to leave. So thank you, and have a good rest of your week.
Thank you. I loved the use cases.
I
loved hearing the use cases to really put it in context.
Oh yeah. I was frantically this is like a whole revamp of my presentation from last year, so I was frantically finishing this one like 30 minutes before sessions. But yeah, I'm gonna start like doing it this way so that people can pick which use case they want to hear, and I'll just keep adding them so as I do more projects. But yeah. All right,
thanks.
Thank you very much for sticking around. I appreciate it. I'm ending the meeting for everyone online. Thank you for sticking around. And let me know if you have any questions.